So we've already seen the general characters of bryophytes, their habitats, distribution. Okay, so we have to move to the next phase where the bryophytes are classified based on their morphological similarities and differences. Okay, the way they reproduce, the way the structure of the gametophyte, the structure of the sporophyte, depending on these characters, they are classified into three chief classes. So normally, commonly, they are called the three classes are called categories are called liverworts, hornworts, and mosses. So these three are commonly known as the bryophytes are broadly categorized into three common categories: liverworts, hornworts, and mosses. ஈரல் புடுகள், கொம்பு புடுகள், மாசுகள் அப்படின்னு நம்ம தமிழ்ல சொல்லுவோம். So we had seen the distribution in the last class, and very common liverworts are Markantia, Rixia, which you have studied in your undergraduates. So in your syllabus, you have Lunularia, which is commonly called crescent cup liverwort. So liverwort, it is, you know the meaning of liverwort because they resemble thalloid portion resembles liver. That's an old comparison. So, so crescent cup liverwort is that gemma cup. It has a crescent appearance. So it is called crescent cup liverwort. So gemma is a asexual, the vegetative plant body which can give rise to a new plant. It's a kind of a vegetative reproduction, or you can call it an asexual reproduction. So here in the inside the gemma cup, you will find many buds, and the gemma cup, when it gets separated from the thallus, main thallus, it can give rise to numerous new plantlets. So that is the shape of the gemma cup here is like a crescent moon, no? Crescent moon you would have seen. So that is why it is referred to as crescent cup liverwort. Another common liverwort example is Rebolia, which is otherwise known as mushroom head liverwort. So most probably the structure, the thalloid structure resembles a mushroom head. So they would have given the common name as mushroom head liverwort. So they are very simple bryophytes having thalloid kind of plant vegetative structure. So they have a thalloid structure. When you come to another category called hornworts, so they would have got this name hornworts because the sporophyte, they arise from the gametophytic thallus like horns, you know. So they must have been named after this. So common hornwort is Anthoceros. You have studied in your UG classes. Uh, so you can see the thalloid structure horizontally over the substratum, and you can see the sporophyte arising out, okay, just like a horn. So another category is mosses. This is very common. You have funaria, polytrichum, sphagnum. I was talking about this sphagnum, which can be found in bogs, that is marshy areas. You can see a lot of sphagnum moss. So it is used in making peat, that is organic manure, for used for horticultural purposes. So sphagnum peat, because it can retain high amount of moisture. So for afforestation, reforestation, everywhere you can add this sphagnum peat. So it will help in the growth of plants. So it has been used in that way. Economically important bryophyte, sphagnum. In your syllabus, you have polytrichum, which is also called commonly hair cap moss. So it must be resembling a hair cap, so that is why they have named it like that. So you have in moss, you have a thalloid structure as well as a differentiated, the vegetative body. So like root-like structures, which are called rhizoids, the axis, the stem-like structure, which is called axis, which bears leaf-like structures. So you have a thalloid structure as well as a foliose structure. And then you can see the sporophytic part of the life cycle, which is attached to the gametophyte. So you can see here also in Anthosaurus also you can see the sporophyte attached to the gametophyte. In moss also you have only difference between the other three, that is liverwort, hornwort and mosses is in moss you can see slightly differentiated form of their vegetative structure into root-like, stem-like, leaf-like structures. So that is a advanced category of bryophyte which resembles the structural aspects of higher plants. Well, it has root like, stem like, and leaf like structures. So these are some examples of the most common categories of bryophytes. So Watson, we follow the Watson system of classification, wherein bryophyta is divided into three classes, main classes. Hepaticopsida. Hepatic means liver. So liverworts are placed under Hepaticopsida. Anthoceratopsida, hornworts, which encompasses the hornworts. It has only one order, Anthoceratales. Okay, and the third one is Bryopsida. Bryopsida, which categorizes the mosses. So under Hepaticopsida, you have these four orders: Calobryaliae, Jungermanniales, Marcantiales, Spirocarpales. Under Anthoceratopsida, you have Anthoceratales. There's only one order. And Bryopsida, you have five orders: Sphagnales, Andriales, Bryales, Polytrichales, and Buxbaumniales. Buxbaumniales. Okay. So this is the outline classification of Bryophyta. Um, you will have to write the general characters of each class: Hepaticopsida, Anthoceratopsida, and Bryopsida, along with the examples to describe the classification of bryophytes. So we will just be seeing small version of each class. So that's what I was mentioning. They are divided into three classes: Hepaticopsida, Anthoceratopsida, and Bryopsida. Hepaticopsida, liverworts. So as I already told you, it comes from the word hepatic, meaning liver. So four orders with their examples are given here. Okay. So when you talk about, when you see the main characters of Hepaticopsida, you already know that the main plant body of bryophyte is the gametophyte. Okay, gametophytic stage is the dominant stage. So the main plant body is the gametophyte. So the main plant body can be a thalloid one or a foliose one. This we have already seen. Thalloid is just attached to the substratum, and one common feature in thalloid structure is it is dichotomous. That means இரண்டாக பிளவுற்ற நுனிப்பகுதி. You can see the dichotomous branching seen very clearly in the thalloid structures of liverworts. See, the tip is always branching in twos. Okay, and leaves here in foliose type of structure, vegetative structure, it is leaf like but without a midrib. So the leaf doesn't have a stalk or a midrib. So you just mention them to be foliose structure. So they are dorsiventral. Dorsiventral means the thalloid forms are also dorsiventral. The foliose forms are also dorsiventral. Dorsiventral means it has each thallus, The plant body has two sides, the upper side and the lower side. Upper side is dorsal side, lower side is called the ventral side. The upper side will have the sexual organs embedded into the thallus, or it will have gemma cups. Okay, the lower side probably normally it has the rhizoids or the scales. Okay, so this is the main plant body of the liverworts. It is either thalloid or foliose, and the leaf-like structure doesn't have stomata. Even though the thalloid structures and the folio structures are green, and they do not have stomatal openings. Okay, and each cell of the thallus, if you see the internal structure, you can see you would have taken section of Markantia in your undergraduate level. So you can see the internal cells will have chloroplast. So normally the bryophyte anatomy is divided into two main parts. The upper part containing chlorophyll, which is the photosynthetic region, and the lower region, which is the storage region. That is how the anatomy is placed in bryophytes. So the cells of the thallus will contain chloroplast without pyrenoids. Okay. So when you talk about the root-like structures, all the rhizoids, they are normally either unicellular or multicellular. They can be branched or unbranched. They can be septate or aseptate. So chiefly in liverworts, you can see aseptate rhizoids. Okay, unbranched aseptate or branched aseptate, and scales are present on the ventral surface. On the lower surface, you can see scales present in the thalloid body, and you can see the rhizoids all along the middle notch portion. Okay, that you can see. And normally the sex organs are bone, they are embedded into the thallus in the gametophytic tissue. When you talk about the liverworts or the hepaticopsida members, the sex organs are not seen. They are embedded into the gametophytic thallus. So the sporophyte is also very simple. It is not showy. You cannot see the sporophyte outside. It is again embedded within the archegonia. So you can see the sporophyte within the archegonia. The previous class I had showed you about the Markantia archegonia 4 and Anthridio 4. So in the archegonia, on the lower surface of the archegonia, you can see the sporophyte developing. It is totally dependent on the gametophytic plant part. So in Rixia, this is a sporophytic stage of Rixia. It's very simple. Uh, in Markantia, it is divided into foot, seta, and capsule. You can see the foot embedded in the inner portion of the archegonium, the seta which comes out, and the capsule which bears the sporogenous tissue. The sporogenous tissue is where the spores are going to be produced. Sporogenous tissue only later gives rise to the spores. Okay, spore mother cells, and each spore mother cell will undergo meiotic division to give rise to spore tetrads. Okay, each spore mother cell will give rise to 4 spores which are attached to each other. So they are called tetrads. Okay, so that is the common aspect in liverworts. The sporophyte is very simple in Rixia. It is made up of only the capsule. You can only see the capsule, whereas in Markantia, you can see the foot. You can actually see the seta and the capsule. Foot is not seen outside. Seta and the capsule. So here also you can see in lunularia, it is in your syllabus, you can see the sporophyte, which is, you can see the seta and the capsule clearly. Okay, it is attached to the archegonia 4 of the particular thallus. And ribolia also you can see on the sporophyte, you can see only the capsule. You can see only the capsule. So this is the characteristic feature of liverwort sporophyte, where in every sporophyte you have some part of the tissue which is very important for the growth and development of the sporophyte. For example, the jacket layer. The jacket layer of the sporogenous tissue, or the layer which covers the sporogenous tissue. Okay, the layer which provides nutrition to the growing sporogenous tissue, columella. Okay, in liverworts, a columella is absent in the capsule. Okay, and the walls are very thin in liverworts. The capsule wall is very thin in liverworts, and you don't find elaters. You would have studied uh, elaters are actually hygroscopic. They absorb water and they swell up, and this is how the spores are released. Okay, so in the capsule kulla vandu you will have spores as well as elaters. Elaters help in the dispersion of spores. Elaters vandu நீரை உறிஞ்சி கொஞ்சம் வீக்கம் அடைந்து, because they are made of thick walls, so they are hygroscopic. So when they absorb water, they swell up, they create a pressure inside the capsule, and due to this pressure the capsule bursts. Either it will burst irregularly or it will burst out through the opening. You will have an opening with a lid, operculum, operculate, in upper operculate capsules are there, depending on the different genera. So in liverworts, you don't find elaters. Okay, you absence of elaters. So there is no special mechanism for dehiscence of capsule. So it's a very irregular way of dehiscence. When the capsule dries up completely, it might just get dehisced through one of the weak points. So the spores are released, and they say you have 2-3 layers which cover the sporogenous tissue. The amphithecium is the outer layer, endothecium is the inner layer. Okay, of the jacket-like covering, no, for during the development of the sporogenous tissue or development of the sporophyte, it will be covered with the outer amphithecium, inner endothecium. So in liverworts, the sporogenous tissue develops from the endothecium, the inner wall of the sporogenous tissue. So these are some characters of the class Hepaticopsida. When you see the reproduction of Hepaticopsida, you can see the gemma cup with a lot of buds inside. Okay, so it takes place asexual reproduction, sexual reproduction is there, and asexual reproduction, you can even call it vegetative. Apart from fragmentation, gemma cup formation, okay, these are the main sources of asexual reproductive bodies. So the buds which are developing inside the gemma cup will give rise to new plant. So sexual reproduction, you have well developed antheridium. See, in Markantia dioecious, you have a male thallus and the female thallus. Okay, so you have anthridiophore bearing thallus and archegoniophore bearing thallus, male gametophyte and female gametophyte. Each one is a thalloid body, call it dioecious. So the male gametophyte will bear the antheridia at the tip. Okay, the tip of the anthridiophore, you can see the development of antheridium, flask shaped structure. This also I was discussing it in the previous class when I was talking about the general characters of bryophyta. You have a flask shaped antheridium. Inside you will have the protoplast will divide into number of androcytes, and each androcyte will give rise to what is called antherozoids, which are called the sperms. Okay, so guess similarly in female gametophyte you have the archegoniophore. On the under surface of the archegoniophore you can see the you can see the venter, okay, near the basal region of the archegoniophore. You can see the venter which will house the egg, and then the neck region. So you have neck canal cells and venter canal cells. Okay, so at a point when the archegonium, the neck region, secretes a certain chemical, and it just comes out and forms a sticky region at the tip of the archegonium, which will attract the sperms which are released by the antheridia, and the the sperm nucleus reaches the egg, and the sporophytic, the embryo is produced. The zygote which is diploid is produced. So embryo, the zygote is the first cell of sporophyte generation. So the sporophyte is developed inside the archegonium itself. So you will have a small portion foot which is embedded in the archegonium, in the venter portion, and then the seta, which is short stalk like structure, which is going to bear the capsule. So inside the capsule you have three layers: exothecium, amphithecium, and endothecium. So the innermost layer only gives rise to the spores in liverworts. The innermost region, that is the endothecium, it gives rise to the spores. The spores are dehisced, doesn't have any regular dehiscing mechanism. When the sporophyte, when the sporogenous, what is the capsule, it dries up, it gets, broken down, the wall breaks down, and the spores are released, and they produce the next gametophytic generation. Okay, so spore tetrad seed spore is haploid, that I had mentioned to you before. So this is the reproduction of liverworts or the hepatic opsida. When you come to the next class, which is Anthoceratopsida, commonly called hornworts. Here also you have just one order, Anthoceratales. Here the plant body is again a thalloid body. Okay, again it is a thalloid body, just like what you saw in liverworts. The gametophytic body is flat, dorsiventral, simple thalloid without internal differentiation. Here also in internal anatomy, you have a photosynthetic region as well as storage region. Okay, and here one difference, when you compare it with liverworts, you have each cell has a chloroplast and pyrenoid. In Hepaticopsida, pyrenoids were not there. It had only chloroplast. But in Anthoceratopsida, members belonging to Anthoceratopsida will have, each cell has chloroplast as well as pyrenoids. Okay, and you can see the internal structure of Anthoceros. You have rhizoids which are smooth walled. Okay, you have, each thallus will bear rhizoids, and again sex organs are embedded dorsally. Okay, on the upper surface of the thallus you can see the sex organs embedded, just like what you saw in liverwort. In Hepaticopsida lercra madriae, ingayum the sex organs will be embedded. You will have the antheridium as well as the archegonium embedded in the same thallus. Okay, and when you have the antheridium producing the sperms and the archegonium producing the eggs, and the sperms swim in the presence of water, they swim towards the archegonium, and everything is the same as what we discussed in Hepaticopsida. Here the difference here is the development of sporophyte is more advanced. So the sporophyte develops into a massive structure which is seen through the naked eye. You can see the sporophytes with the help of your eyes, because it arises from the gametophytic thallus. Okay, as a horn, looks like a horn arising from the dorsal surface of the thallus. So it is differentiated into a well deep-seated foot. You have a long seta which bears the capsule. This is a very important, examination point of view, a very important question. Discuss the sporophyte of Anthoceros. In your UG, you would have studied in detail the structure of the sporophyte of Anthoceros. It's very complex and very structurally very well differentiated. Here, the sporogenous tissues develop from amphithecium, from the middle layer of the spore wall. Okay, so in Hepaticopsida, you saw the sporogenous tissues, the spore mother cells, they develop from the endothecium. Here, in Anthoceratopsida, the spore mother cells or the sporogenous tissues develop from the amphithecium. And you find elater-like cells. You can call them elaters because you have they have thickenings around the elater. They are referred to as pseudo-elaters. They are hygroscopic, but they don't take up the full function of the elaters. Okay, and then you have columella, small region of columella you can see in the sporogenous tissue in the capsule you can see, which originates from the endothecium. So there was absence of columella, that is Hepaticopsida members, they don't have columella, whereas Anthoceratopsida, they have columella which is originating from the endothecium. Sporogenous tissues originate from the amphithecium. So you can see the spore tetrads just surrounding the columella. You have the sporogenous tissue which is going to give rise to the spore tetrads. And you have, there are many stomata, openings are there. Okay, stomata like openings are there throughout the thallus. This is another difference from the Hepaticopsida where there was no stomata seen in the thallus of Hepaticopsida or the liverworts. In Anthoceratopsida, you have the presence of stomata, you have the presence of columella, you have pseudoelaters, you have pyrenoids. See, you have the well differentiated sporophyte into foot seta and capsule. Okay, so these are all some major differences between Hepaticopsida and Anthoceratopsida. Then we will move on to the reproduction seen in Anthoceratopsida. Apart from fragmentation, apart you have some species also show gemma cup, like Anthoceros glandulosus. It has a gemma cup. Okay, and apart from that, you have tubers which are developed in the tips of the in some places in the thallus. They have an outer protective corky layer. Okay, and they tolerate extreme dryness because these bryophytes they require high moisture for growth and development. But during unfavorable condition, when there is a lot of dryness in the environment, வறண்ட சூழல் இருக்கும் பொழுது you have the production of some structural parts like tubers. Okay, and the tubers are actually covered by cork. Okay, cork, so which helps the plant to survive the dry condition. So even if the thallus is not able to survive, the tubers will survive. So on the return of a high moisture content in the environment, the tubers will give rise to the new thallus. So here in Anthoceratopsida, you have vegetative reproduction, asexual reproduction by forming tubers. So this is formed only during unfavorable conditions. Okay, in under favorable conditions, it will reproduce asexually by fragmentation or gemma cup. Only during unfavorable conditions, Anthoceratopsida members will show, some of them will show tuber formation. The sexual reproduction is same as what we discussed in Hepaticopsida, where you have the antheridium and the archegonium bone on the same thallus. Bone on the same thallus. In Markantia, we have seen male gametophyte and female gametophyte, but here you can see in the same thallus, you can see the development of antheridium and archegonium, and they develop they are both embedded in the thallus. Dorsally, they are both embedded, and the rest all is the same as you have seen in Hepaticopsida. So when you come to the third class, Bryopsida, which is commonly called the mosses. Okay, you have the main feature is you have you have a thalloid portion in the plant body, which is called protonema, okay, which will grow horizontally over the substratum. Okay, they may be branched, they will contain rhizoids, they will be branched. Okay, and then you will have a foliose region also. Okay, so the thallus in Bryopsida is having two parts: horizontal protonema and an aerial foliose part. So it is quite differentiated into root-like, stem-like, and leaf-like structures. Okay, so gametophyte is a protonemal, thalloid protonemal plus a foliose gametophore, you call them. Okay, so foliose is made up of a stem. You call it axis actually. It is not called stem, it is called axis. And you have leaf-like structures. The leaves are without midrib portion. Okay, and rhizoids are multicellular with oblique septate rhizoids. Okay, in the Hepaticopsida and Anthoceratopsida, you don't find septate rhizoids, whereas in Bryopsida, you find septate rhizoids.